My name is Professor Arif Herekar. I'm basically a professor of both adult and child neurology. I have a master's in child neurology from the University of Rome, done about 8 years ago, and I have my MD in neurology. I've been the ex-chairman of the neurosciences at the Bakai Medical University in Karachi. I thought with 40 years of exposure to child and adult neurology, I thought of passing this knowledge on to the youngsters who are inclined towards or who have a passion to learn pediatric or child neurology. Hope you will learn from these sessions and you will carry back home more information than you had before the session. Thank you. In utero, the critically affects the brain development. Now, the worst time that a child's brain can be traumatized is when it's developing in utero. Thereby, it modifies the neurobehavior of the offspring after birth. Substances that are ingested by a pregnant mother or woman can cross the placenta and adversely affect the fetal development. So this is where things start to disrupt or slow down. Some are unavoidable medications, not all are avoidable, due to the mother's medical condition. She may have any medical condition, such as epilepsy or depression. Diabetes, of course, is a very, very important condition. And there are quite a lot of diabetics, at least in our country. But the others are consumed by an expectant mother, unbeknown to the risks and effects. She continues to take medication. She does not know how bad it can be. In some cases, an excessive amount may be taken by a pregnant woman due to addiction. So this is one thing. Addiction can badly affect the growing brain in utero. During brain development, newly generated neurons, everything happens in neurons, undergo morphological changes, followed by migrating from the germinal layer through the intricate network. Of course, there's a huge network of neurons in the brain. And this intricate network of extracellular matrices to establish connections with other cells in the highly ordered fashion. Everything is in order in the brain. Any disruption, any variation, any diversity is going to cause a disease process. Two major modes of migration in the cerebral cortex are known. The first is radial migration. Now, excitatory neurons generated in the proliferative zones of the dorsal telencephalic primordium radially migrate toward the PL surface of the cerebral cortex along the radial axis. I'm sure we'll be describing that diagrammatically as well. You can see that. These neurons use the cellular processes that are elongated from the radial glial cell. So these are like elongated processes which are ascending neural progenitor cells as the scaffold of their migration. Now, having said that, on arriving at the superficial layer, the neurons detach from the radial glial processes to settle down to what we call the cortical plate. Another mode of neuronal migration, known as tangential migration, is employed by inhibitory interneurons. So now we are talking of the inhibitory interneurons. These neurons migrate tangentially from the ganglionic eminences, which are called GE, to the cerebral cortex. Now, any disturbance of these two modes of migration can cause what we are worried about, the neuronal migration disorders, what we call NMDs. And the consequent malformations are detectable by brain imaging in NMD patients. Profound cases of NMDs include now lissencephaly, number 1, heterotopia, number 2, and focal dysplasias. Dysplasia means something that has not been formed properly. Precise control of the migration and positioning of both the excitatory and the inhibitory neurons are particularly important for the formation of course, we are talking of the synapses. So these are very, very important, and they are important for the formation of the synaptic excitation and inhibition. We are talking of both excitation and inhibition. Balanced circuit in the brain. Therefore, the imbalance is the main cause of epilepsy in patients with neuronal migration disorders. Other neurodevelopmental and psychiatric disorders can also occur, which include, like we are encountering so many patients with autism, the autism spectrum disorders, which you call ASDs, and of course, schizophrenia are also associated with NMDs. Now, in addition to the genetic causes, harmful prenatal environment also leads to neuronal migration defects. When developmental neurotoxicity first emerged, various paradigms of exposure were deployed in many species to recapitulate human pathophysiology, including defects of neural migration. Now, decades later, now, many recent studies have utilized genetic tools and cutting-edge molecular techniques. Now, obviously, the new techniques are coming in. And these are employed in those preclinical animal models to decipher the underlying mechanism of neuronal migration disorders under stress exposure. So I hope this first part of my child neurology or pediatric neurology lectures you have understood. This is just the beginning, this is just the introduction. We'll continue to bring more such sessions so that the basics of child or pediatric neurology is understood and clarified. Do subscribe to my channel and hope you have enjoyed it. Write a little comment. You know what the comment does is it improves your next video. And of course, if you like this video, press the like icon. Thank you so much.